This YouTube series is designed for upper year undergrad biology students in order to help them learn about the use of CRISPR-Cas9 technology in breast cancer treatment, along with the limitations and challenges associated with this method. To begin, let's first establish what breast cancer is. Breast cancer is defined as a heterogeneous disease in which malignant cells form in the tissues of the breast. In 2020 alone, 2.3 million women were diagnosed with breast cancer and 685,000 deaths were recorded globally. Moreover, 1 in 12 women are expected to develop this disease during their lifetime, making breast cancer one of the most prevalent forms of cancers worldwide. The majority of breast cancers start in the glandular tissue of the breast and are referred to as adenocarcinomas. In most cases, classification is based on tumor origin, either in the ducts, called ductal carcinoma, or in the lobules, known as lobular carcinoma. Ductal carcinoma is the most common type of breast cancer, accounting for about 85% of all in situ breast cancers and 70 to 80% of all invasive forms. Additionally, there are some less common but more serious forms of breast cancer, including HER2-positive breast cancer, triple-negative breast cancer, inflammatory breast cancer, and Paget's disease of the breast. Once a biopsy is done, breast cancer cells are tested for proteins called estrogen receptors, progesterone receptors, and the human epidermal growth factor receptors. The specific proteins identified help in determining the treatment options that can include surgery, radiation, chemotherapy, hormone-based therapy, targeted therapy, or immunotherapy. The type of treatment received is based primarily on the breast cancer classification and stage, as well as the patient's personal preferences. Since cancer is caused by mutations in the genome, a specific gene editing tool called CRISPR-Cas9 can be used to correct causative mutations with minimal toxicity. The CRISPR-Cas9 system can be employed to virtually edit any segment of DNA within the 3 billion letters of the human genome, and it is more precise than other DNA editing tools. Moreover, it has certain advantages over the traditional forms of treatments. Currently available therapeutic agents are associated with high toxicity levels and low success rates. Additionally, ongoing multi-targeted therapies are limited to few carcinogenesis pathways due to continually emerging and evolving mutations of proto-oncogenes and tumor suppressive genes. CRISPR is short for clusters of regularly interspaced short palindromic repeats. This genome editing tool consists of a pre-designed RNA sequence, which is known as guide RNA, and a CRISPR-associated endonuclease known as the Cas9 enzyme. Guide RNA sequence is a short palindromic RNA sequence with bases that are complementary to target DNA sequences. Therefore, the gRNA sequence is restricted to only binding to the target sequence and not other regions of the genome. With the help of gRNA, the Cas enzyme is directed to a specific part of a gene and initiates a cut across both strands of DNA in the proximity of the protospacer adjacent motif. Once the CRISPR system has made a cut, the double-stranded break can be repaired either through error-prone non-homologous enjoining or a homology-directed repair pathway. In the non-homologous enjoining pathway, the ends of double-stranded breaks are processed by endogenous DNA repair machinery and rejoin, which can result in random indel mutations at the junction site. Indel mutations refer to insertions or deletions of nucleotides within the coding region of genes, which results in a frame shift or creation of a premature stop codon. In the homologous enjoining pathway, a repair template in the form of a plasmid or a single-stranded oligodeoxynucleotide is supplied to knock in the gene of interest into the genome via accessory factor. Overall, both DNA repair pathways result in a desirable genome editing outcome. However, the homology-directed repair pathway generates precise genome modification at the target site in comparison to the non-homologous enjoining pathway. In this video, we will discuss the potential use of CRISPR-Cas9 genome editing tool to treat various forms of breast cancer, including HER2-positive breast cancer and triple-negative breast cancer, as well as explore CRISPR-Cas9's use in CAR T-cell therapy.